Brian Wandell, Stein Family Professor from the Department of Psychology at Stanford University. He obtained his bachelor in mathematics and psychology from the University of Michigan and his PhD in social sciences from the University of California, Irvine. He spent a year as a postdoc at the University of Pennsylvania, and then he joined the Stanford University. He's also the director of the Stanford Center for Cognitive and Neurobiological Imaging. His research centers on vision science, spanning topics from visual disorders, reading development in children, to digital imaging devices and algorithms for both magnetic resonance imaging and digital imaging. So it's a wide spectrum of interests. So welcome to Boulder.
Thank you. Thanks, Ariel. Yeah. Thank you. And thank you for organizing such a nice visit and such a nice day. I appreciate it very much. I'm going to go a little quickly through material, but I'm going to be looking at your faces and counting on you to stop me when you look a little confused. I'm going to slow down, and you'll tell me how I'm doing here. I do understand that this is very different. How many of you woke up this morning thinking about the brain and brain science? Okay, well, that's actually more than I thought was going to. and I assumed that, and it seems roughly right, that most of you are engineers or physicists, and I was going to start out with a little background about the brain and brain parts, and just a little stuff to get the terminology right and to try to make you comfortable with the ideas that I'll be talking about later. I will then talk about work that I did for a long period of time, from the early 90s till about the 2005, 2007, using one kind of brain imaging called functional magnetic resonance imaging. Magnetic resonance imaging is great because it has many different modes in which it can be used. And I'll be talking about work on functional magnetic resonance imaging to help clarify in the human brain how the visual parts of cortex are organized. And I'll usually vision science, because vision is such a big part of the human brain, and because there are so many vision scientists, a lot of ideas that get started in vision science end up migrating into other parts of neuroscience. And so I'll be explaining what our thinking was then, and how things are evolving, and how that might end up applying to other parts of the brain. Then I will shift about halfway through into another mode of using magnetic resonance imaging called diffusion magnetic resonance imaging. And this is a mode in which we are learning about structures in the brain that connect between different neurons at a large scale. So diffusion magnetic resonance imaging and tractography. And by then you will all be experts in the brain, functional MRI, and structural MRI. So I'll give you a little bit of sense of the kinds of applications that we might use based on those techniques to understand. I have two things in mind. One is a little bit about reading, which has been studying how children learn to read, and the parts of their brain that develop during reading has been a big focus for my lab for about the last 7 or 8 years. And I'll give you just a little sense of that. There are many papers, but I'm here to give you a sense of that. And then how this world continues to work with surprising phenomena. And I may not get to the surprise, but if I do, good. If I don't, ask me about it at a beer afterwards. Okay, so first off, the parts list. This is a lateral view of one side of a human brain. Brains across different species are wildly different. When you go across to the biology department, they rarely know about human brains in biology departments. They know everything that there is to know about the mouse brain, a fair amount about Drosophila, nervous system, and so forth. Human brain, they leave that to psychologists, by and large, or people in the med school. And it's painted into different regions, and really the segmentation into these different regions, like the occipital lobe, which I'll be talking about a fair amount, or the parietal lobe, or the frontal lobe, or the temporal lobe. These different regions are just anatomical conveniences. You kind of look at a brain, you see a deep fissure that divides two parts, and you say, well, let's call this part the occipital and this part the temporal, and it's a way of just sort of navigating, kind of like north, east, west, and south, that kinds of stuff. You can roughly assign functions to different parts. So this part back here, occipital lobe, where we'll be spending a lot of time, is very responsive to visual stimuli and can be driven in very orderly ways in response to particular visual stimuli that I'll be showing you about. If you have lesions, and we've really relied on neurologists over more than 100 years now to tell us about this, if you have lesions in particular parts of the brain, these lesions are manifest in the behavior of subjects by very specific, sometimes, not always, but sometimes by very specific losses. So, for example, a lesion back here typically leads to blindness. A lesion over here will lead to not blindness, but perhaps to the inability to recognize faces or to read, depending on exactly where this thing is. And it's not the case that every piece of brain has an associated function that we understand, or is it the case that every function will be assigned to a particular piece of brain? That's the problem we're supposed to work out, how these computations are done. But for some functions, some pieces of brain are absolutely essential. So back here, if this bit isn't working properly, you will be blind. It's essential. It's not the only part needed to see, but it is essential. Without it, you won't see. Now, this is the kind of thing that we teach in introductory psychology, and I think it's a good thing to start with and to learn about. I would also say that as you move forward, the kind of grip on neuroscience that we teach in graduate programs and so forth is pretty much focused on the computational elements of the brain, which are the neurons. And if you look at an MR scan, here's a slice through the brain over here. So this horizontal line refers to where this slice is. Here's the back of the brain. Here the eyes would be up over here somewhere, up over here. And in a MR scan like this one, a little piece of the brain like that, let's say a cubic millimeter on a side, has within it about 50,000 neurons. And the neurons are the computational elements, there's the cell bodies, and the places where, here's a cell body, of a particular neuron, and then they will have coming in and out of them. Well, you could have this piece.
Here's that. I'll take this one.
Okay. That was nice. They have inputs and outputs near the cell body. That the inputs are the dendrites that receive signals from other neurons, and outputs are the axons that carry the outputs after the information from the dendrites has been accumulated and changed, transformed in some way. A decision is made as to whether a neuron is going to send the signal on out, and it'll send it out to the axons. And there are many types of neurons in a little chunk, cubic millimeter. There's 50,000 total, and in visual cortex, there's probably about a dozen different types that we know about already, and likely to be many more types. Now, one of the things that's really fundamentally different, it's great, I really enjoy hanging out in schools of engineering because they seem endlessly interested in the way the brain does computations. And can we learn something to do something at lower power? And so I'm always interested in comparing the characteristics of integrated circuits with the brain. And one of the things that's really different is that each neuron in the brain typically contacts between 1 and 10,000 other neurons. So the density of interconnects is really massive compared to the density of interconnects in ICs. The other thing that is maybe similar, but it's hard to tell, is that we're zooming in here looking at all these individual neurons, but some of them actually send their outputs into the white matter over here. So this region on the surface over here on the outside is called the gray matter because it looks kind of gray when you look at it. And the stuff in the middle is called white matter because those are the axons of these cells that are sending their outputs to other neurons, sometimes quite remotely. And for understanding this circuit, we need to understand both the local computational elements and where they send their signals to. And that's what will come up here. So let me zoom in on a particular, the other thing, by the way, I'm guessing as engineers, you would focus on this also, that when you see a set of neurons that look like this, and you see a neuron that looks like this, you think, wow, they probably have different functions. And that's not a fundamental idea in neuroscience. I don't know why. Some people kind of give it some lip service, but the idea that these would have really different functions is something that's obvious to an engineer who looks at the structure, say, gee, I bet that structure really matches something that's different. In fact, most of single unit neuroscience involves poking needles into the cortex, measuring the activity of a cell, and not even knowing whether it was this type or this type of cell. That's the overwhelming majority of single unit recording. But I think that will change as technology evolves and computations can start thinking about that right away. Actually, let me move along. Again, I just want to emphasize this is probably good enough for what we need to know, that the inputs come in here through the dendrites, the cell body is here of a neuron, and the axon is the output, which then goes in synapses on another cell. And this is drawn for one synapse in one cell, but a typical cell like this will synapse at 1,000 other cells. Yeah.
So are you saying, because on the last slide, different types of neurons, are you saying that they do have different?
Almost for sure.
Almost for sure?
Almost for sure.
Okay.
And you can tell the neuroscientist that for me. No, actually, there are many, many who would agree with me, but I would say that it's not in their. The clearest example is, for example, the retina, in which all the output cell types have at this point been identified. There's about 20 different types that you can distinguish in the retina like this. And they actually send their projections to different places. They have different electrical properties. They have different transmitter systems and so forth. And I always use the retina as kind of the model system for the way the rest of the system will be understood. So yeah, I'm sorry, I'm being a little, if I thought this was a group of square-on neuroscientists, I would be a little more polite in my description. But I'm going to be a little flip, even though I love neuroscientists and all that they've done, and I'll tell you some of that as we go on. Okay, so the surface, when you look at a brain like this, the surface is about 2 to 5 millimeters thick, filled with these cell bodies, and that's what's called the cortex. Well, this part over here. There are several other major structures, but the part that you all would normally think about, the neocortex, the new cortex, sits up there. And it's a thin sheet, 2 to 5 millimeters thick. It's packed with 50,000 cells per cubic millimeter and so forth. And the neurons in there are impulse conducting. They're the little electrical generators, cells in here. And their outputs are the axons, a thin fiber that carries the output impulses from a neuron. Some of them spend their whole lives talking only to their neighbors, just locally, nearby. And on occasional, some of them, their axons go into the white matter, forming up the white matter. And it looks white because their axons are surrounded by this fatty tissue. It's a lipid called myelin. Everything's lipids, but in particular the myelin that wraps these things that helps them conduct in an efficient way. And so they go out, and then the point of contact between neurons is the synapse. And these are rough numbers, almost a rough order of magnitude numbers, as I said. Neurons per cubic millimeter, 50 to, there are parts that are more dense. Total number of neurons, probably on the order of, cortical neurons, probably on the order of 50 billion, 100 billion. Synapses per neuron, about 10 to the third. Total cortical in the trillions. And each hemisphere, they're very folded. The human is very, it's called, is filled with gyri and sulci, very, very folded for the human. Other animals have smoother brains, but we have a lot of cortical territory buried into these folds called sulci. And if you take can I just borrow this for a sec? If you take one hemisphere and flatten it out, it's about the size of a sheet of paper, like that. You can see why it would be folded, because if your brain on each side were that flat and you kept it flat like that, it would be a problem getting through doors and stuff like that. The whole birth process would scare the hell out of me. Okay. Okay, so here's a picture of the wiring. Here's a picture of the wiring, the synapses, and so forth. And this is really my favorite number, that if you take a cubic millimeter of cortex and you take all the axons, all the outputs between the cells in a cubic millimeter, and you just stretch it out, it goes for 3 kilometers. So this connectivity stuff is really, really important in the human brain. Okay, so that was very hard. All that was hard-fought information delivered by biologists and physiologists in the 1880 to 1940 time frame, and some of it more modern. The rise of functional magnetic resonance imaging is much more recent, but it starts really with an observation made a couple of different groups. You can tell the story in a number of different ways. This isn't historically pure, but it is one of the threads. There's nothing wrong. It's not the complete story. I particularly like this guy's paper, J.F. Fulton, writing in 1928. He was a medical doctor writing out of Boston, I think out of Harvard actually. And he had a patient, Walter K., come to him. And Walter was a sailor. And Walter, throughout his life, had been blind in half of his visual field. And in fact, in the right half of his visual field. And for those of you with a little familiarity with the visual system or neurology and so forth, the left hemisphere receives its input from the right part of the visual field. Everything you fixate at something, everything to the right starts out in the back of your left. When it gets into your brain, it ends up in the left side over there. And you'll see that many times as we go forward in these slides. So he had been blind over there, but now things had started to hurt. And so he came to the doctor and said, "Doctor, it hurts. That's why you go to the doctor." And Fulton, and it bothered him a lot. And Fulton said, well, in the 1928, we didn't have MRs. There wasn't a lot you could do. So if it hurt so much that it really was a problem, the only thing you could really do is crack open the guy's skull and have a look. And so that's also called surgery. And so this is how he wrote about it. He said, "I'm turning down a left occipital," so now you know occipital back here, "bone flap, a large, angry-looking angioma," and they do still write this way, angry-looking or severe-looking and so forth. Angioma arteriolar racemosa, means of the left occipital lobe, was disclosed, which extensively involved the visual cortex, right? The occipital lobe. It's also called visual cortex sometimes. The hemorrhage, the bleeding occasioned by the bone flap, we opened it up and blood just started to spurt everywhere, was so excessive that the operation had to be abandoned without touching this bad stuff that had presumably blinded him in there. But a decompression, in other words, we'd let a lot of blood out and a lot of liquid came out of there, and we put his skull back on, and he kind of woke up and he said, "I'm feeling a lot better, doc." And they said, "Great." Patient was discharged, less pain, it was just better. Okay, now he went back out to sea. And back then, when you put somebody's skull back on their head, they didn't tighten it down quite as well as we could now. So there was not a big crack, but just a little, auditory signals could come through there and you could hear stuff. And Walter K. would be out at sea, and he'd be looking to try to see a star or something. And whenever he would look hard to try to see something, his mates would hear psh, 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 coming out of the back of his skull. And then, when he wasn't looking hard or trying to see anything, just, close his eyes or just, whatever, they wouldn't hear anything. So he came back to Fulton and said, "Look, I'm feeling a lot better, but I got this noise coming out of my head. What do you think about that?" So Fulton took out his stethoscope and actually recorded the waveform and put it down on a little trace, and that was the basis of this paper. And he said, well, look, try to read that fine pattern, like you got pearl streak there on your, try to read this stuff. And when he tried to read it, it went psh, 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 psh. So, well, now listen for a ticking clock. Nothing. Or count backwards from 100, and he'd count backwards. Nothing. Say, okay, look, can you see the detail back over there? Psh, 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 psh. So Fulton correctly concluded that blood was delivered to the part of the brain that was active at that moment in time, and that the flow of blood was in fact distributed in a way that reflected your mental processes, be they vision or hearing or listening or thought and so forth. And that's important because that's actually at the basis of functional magnetic resonance imaging. And in fact, there's a lot to go beyond this. I teach an entire class devoted to the MR physics of this, and I'm just finishing it now, so it's all fresh in my mind. There's a lot to go, but I'm going to give you 1.5 slides worth on this, which the basic thing that was worked out by Sokolov, Mark Rako, Peter Fox, and so forth in the 70s and 80s is that the heart lung oxygenates the blood that is going to go to the brain. And in the arteries, you get basically 100% oxygenated blood headed throughout the brain. When there's a region of the brain that's active, let's say in the control state, you're just sitting there relaxed, not doing much, about 40% of the oxygen is extracted and 60% not, so that on the return, you get a 60-40 split. Now, when you have somebody look very hard at something or do a difficult task, in fact, that piece of brain demands more, is going to use more oxygen because it's going to metabolize the ATP and turn it into energy that it's going to use, and it demands more oxygen. So you can see we've gone from whatever this unit is to 30% more over here. So it gets more oxygen. It in fact uses more oxygen, but not so much more as what it got. So on the return trip in an active state, you actually, instead of being 60-40, you might have something like 63-37, something like that. Now, that's from the point of that difference, 60-40 versus 63-37, is the basis of the difference that we pick up in a functional magnetic resonance imaging experiment. And the reason we can pick it up is because the magnetic field properties, the magnetic properties, diamagnetic versus paramagnetic, is a difference between oxygenated and deoxygenated blood. Okay, so oxygenated blood is transparent to the magnetic field, and deoxygenated blood has a bit of susceptibility, changes the local magnetic field a little bit. And so in MR scanners, what we're looking for functional scans are differences in the relative blood oxygen level. And Fulton's observation that the amount of blood rushing to one part of the brain versus another depends on what person's doing, coupled with something that started with Linus Pauling and then a whole series of other important findings, says that in MR scanners, we can actually go pick up these differences in a spatially localized way. So what we get in fMRI is this hugely indirect measure of the actual neural signal. And we don't know whether it's one type of cell or another type of cell. We don't know exactly which is the energy-consuming part of the response that draws this blood. So there's a lot of things we don't know. But on the other hand, we can take a perfectly healthy, living undergraduate and put them in a scanner, and without hurting their skull or cracking them open or anything, measure at the resolution of about a cubic millimeter or 2 cubic millimeters on a side, we can measure the blood flowing in and out of their brain without hurting them. None of that. We give them $40 and send them off to go and do whatever they do with $40 these days. So it's kind of cool. And we can visualize it and measure it, and we can do it, and we can show them all kinds of things and do all kinds of experiments to get access to the functional properties of the living human brain inside of an MR scanner, which they could have done for Walter Kay back in 1928 and learned a lot more, though it might not have rescued his vision. Okay, so what's the first thing? So the first MR scanner shipped in the United States in 1982. In about 1990, Seiji Ogawa, who was working at the Bell Labs, David Tank, Camille Ugerbill, a number of people, realized that they could use these MR scanners and observations about the relative susceptibility of oxygenated and deoxygenated blood to make measurements in the human brain. And in about 93, 94, Ken Kwong at Harvard showed that, in fact, you could see the activity in the human brain with these kinds of MR scanners. And right at that moment, our group said, well, okay, look, let's see what we can do. But there was actually some controversy at the time about whether or not what the signal was. And we got into the field about that time to try to study visual cortex because my background had been in vision science and visual neuroscience. So this is a picture then, the right hemisphere of a human brain. And this area over here had been known for 100 years before we got into, well, almost 100 years from before we got into it, as where primary visual cortex is located. The purple stuff is occipital lobe and parts that are responsive to visual stimuli in general. The first place where signals arrive in cortex is shown here inside of this little sulcus. I'm going to zoom in on it here, called the calcarine sulcus. And from neurologists, we had known that it's organized, this little region of cortex is organized into a map that roughly follows the visual field map. And what I mean by that is this is the right hemisphere of the brain. And so nothing over here if you're focused here, nothing on the right side actually drives this. It doesn't get there in the first step. But the left side, the opposite, the left visual field will drive the right hemisphere. And things near the fovea over here, in the center of your visual field, will drive cells over here at the back. And as you go increasingly eccentric, it will drive activity increasingly anterior along this. If you look at the vertical meridian, the lower vertical meridian will drive cells that are active up over here, and the upper vertical meridian of the visual field will drive cells that are active at the bottom over there. So it's like a little map of the hemifield over there. And so the first thing we did, because there was a bit of controversy about how well you could spatially localize, in vision we didn't have to say, well, gee, I wonder where visual cortex is. We knew where visual cortex is. So we could use this system as a little model system to measure the resolution of this technique. And these are what the data look like. So if you focus here, you can see here's an expanding ring. I'll just let it play for a while and give you a chance to look back and forth. You can see that the activity, when it starts out in the center, the activity starts at the back. And as this ring expands, you can see it travels, the activation travels forward. This is also the visual cortex. And what we've done here, what we did in the early days was segment it out and expand it out so you could see down into the sulci. The dark shaded parts would be down in the bottom of the sulci. You wouldn't normally see it, but since we puffed up the brain a little bit, you can now see the entire pattern of activity. And you can see the eccentricity map from the fovea to the periphery in the traveling wave of activation in primary visual cortex. And so that gives you a sense of how well you can localize stuff. There's other things that are going on down here. You can see this activity down in here. We're going to come back and study this piece of cortex later and when we get to reading and so forth. But there's other stuff going on down here. Notice it starts out and then it kind of weakens as the stuff gets in the periphery. In fact, that's true. This piece of cortex down here is particularly responsive to things in the center of your visual field, what's called the foveal region in the center where your fixation is and so forth. And the nice thing about this, you can do this. We were very excited when we first started to see this in 93, 94 and so forth. It was particularly Steve Engel in my lab and Marty Serino, Ted DeYoe, Roger Chutel and others. We were all doing this competitively as a competitive sport in 93, 94, 95. And by now, it's a completely standard thing. You're never happy when you see this. You're only unhappy when you fail to see it. And that's, I guess, the tragedy of being a scientist. So you can represent these in a static view by using a color map. So if red is the foveal activation and green perifoveal, and then the further periphery, these blues and purples, you can see that it lays out roughly like this from the fovea onto the periphery. Now you can do another experiment for the other dimension. You can take a wedge like this one and just have it go around in a circle, and that'll get you the other dimension. And you can see this blue and red over here. The blue maps out over here. The red maps out over here. This is the upper lip of the calcarine, the lower lip of the calcarine. And then as you look at these, if you just take a moment with me, you can see it goes from red to green to blue like this. And then it goes from blue to green, and then back to blue. That's a very regular pattern. Very regular. And so that issue and several things like that had us gripped for many years, because we knew that in animal models there was more than one map, not just the V1 map, but additional maps. And here for the first time we could see the additional maps in human visual cortex, and neurologists hadn't been able to segment out the second visual map and the third visual map and so forth. And so that really gripped a number of us from about 93 to 2003, 2005, to try to find as many visual field maps as we could in human visual cortex to lay out the basic architecture. And so we took those two kinds of experiments, and I guess by about 2007, which is, I don't know if this one comes from the I think this comes from the 2007 paper, we had about 16 visual field maps that had been reported by at least 2 groups. There's more that have been reported, but not, it's like any other field. Sometimes they're right, sometimes they're wrong. But by at least 2 groups that agreed on at least 16. I think it's up to over 20 at this point. So that was 5 years ago. But the pace is slowing down. So it's one of those curves that's now a compressive function that's basically turned over. And we've set up some reasonable naming conventions for these things and so forth, and I can tell you about that. But that part of the work, nearly the entire occipital lobe is tiled with these maps, and there's a whole bunch of them, and we know things about their organization and structure.
How big are those regions?
The biggest one, the best numbers we have are for V1, V2, V3 in a paper by Doherty in 2003 or something. And from here to the most eccentric part is about 8 centimeters. And from the upper to lower for V1 is about 4 centimeters. So they're pretty hefty chunks of folded cortex. And as you go to some of these more anterior ones, they could be on the order of 2 centimeters on a side in the human brain. The ones that are homologous to these in animals, such as macaque, you got to divide each dimension down by at least 2, maybe more, so that they're about an area about a factor of 4 smaller. In mouse, they're another order of magnitude. They're just much smaller. And the ability to make comparisons between species is really limited. So it's cool that we can get these things in human. I can tell you that the first really big fight I got into, it's been a kind of a pleasant field by and large, but the first tense argument I had about these maps was the 4th visual field map in human has an organization and position that's not much like the 4th map in monkeys. And the in macaque monkey. There's a lot of monkeys out there, but in macaque. And the debates about that were heated for a while. I'm pretty convinced we've won that argument, but that's another, I'll let you read the literature. That's summarized in part in that paper. A couple of centimeters on a side, different in human from even macaque, quite different in their layout and probably function from lots of other species. Okay, so having the maps is a start, but another thing you'd like to know is what happens in those maps, and you'd like to measure the properties of these masses of neurons. And the key idea that we've been using in recent years, since about 2007, 2008, has been an idea drawn from the physiological literature to look at each of the voxels and try to understand, when you look at some particular piece of cortex, what part of the visual field would excite that particular location in cortex. This would be called the receptive field. It was an idea introduced by H.K. Hartline in 1936. Responses can be obtained in a particular cell or optic nerve fiber only when you illuminate a small part of the visual field. And mostly a particular cell won't see other parts of the field. And that's true for, these maps tell you that's true also, that the cells at one place sees this one part of the field but not the other part. So let's measure that. Let's see what that is and see how that changes across these maps. And that starts to involve a little bit of computation, a little bit of modeling. And let me just take you through it. Suppose we had one of these stimuli, like I showed you this rotating wedge stimulus before, and I had a particular voxel, a particular location in the brain, and it had a time series shown here in blue as this thing rotated around through a cycle, nothing, 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 and then whoop, it starts to go off, and then nothing, 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 like that. And I started out and I said, let's suppose that its receptive field was roughly circular and was located over here. I could actually make a prediction of what I should see over here, and it would be this black curve. And I'd say to Raphael, how'd I do on my prediction? And he's a hard-nosed kind of guy. He'd say, boy, that's a bad prediction. I'd say, okay, right? Yeah, okay. So let's move it over here. Now how am I doing on my prediction? And some of you would be generous and say, oh, you're doing very well, But Raphael, he's a tough-minded guy. He says, it's still a lose, because look at this black thing comes up and down, and this thing's still going, so that's no good. Okay. So you say, well, what if we make it bigger? Now everybody's happy. Right? So from this kind of so you can see, obviously you do this with the right minimization rules, and you do it of lots of voxels, and so that's the basic idea. You take a model of what you think receptive fields look like. You have parameters, the central position and the spread in this case, and you place them here in order to minimize the deviation between the prediction of when the stimulus excites the receptive field and the observed data coming from that voxel.
It seems like time constant between excitation and observation should be of interest.
Very important. And in this field, that's a great point, and I was going to sail over it. But that's right. That's called the hemodynamic response function. And it's basically an impulse response function that tells you when the stimulus is at your eye, there's a certain time course for the response. And in this field, it's very slow. So the time to peak is about 3 to 6 seconds. So to go and make this kind of a calculation, you also need to know something about the hemodynamic response function, which I'm not taking you through, but it's urgent to take that, to do that the right way. So that's in fact part of our estimation process. So that's true and a very good point. But you can also, so anyway, once you do that and do a bunch of things like, there's other things wrong with what I've told you here also, or incomplete at least about what I've told you. Once you do that, you can look at, let's say, this piece of visual cortex. This is the middle surface, and here's primary visual cortex. And here's a part that's responsive on the lateral surface, and you can look at the size of the receptive fields in these two parts that I started out just telling you were parts of visual cortex, and you can see, wow, their time series were really different, so that the fit that I needed to explain this particular voxel or this particular voxel, the fit I needed was very different. And in fact, this we call the size of the population receptive field, and we can measure it for every one of these purple points. Its center and its position. And we have lots of aggravation about the quality of measurements and so forth, but it's not so bad. And we can look in, for example, V1, V2, the second map, the fourth map. We can look in all these different maps, and even within maps, if you look at the size of the receptive field, so that this axis here is the size of the receptive field, and the center of the receptive field in terms of whether it's near the fovea or eccentric out in the periphery, it changes systematically as you go from the fovea, where you have small receptive fields and therefore high resolution, to the periphery, where you have bigger receptive fields and lower resolution in very systematic, orderly ways. And as you go from one map to the next map and so forth, they also change so that these maps out here, these are motion responsive maps, temporal occipital 1, temporal occipital 2. These maps here have giant receptive fields, and these maps here, the earlier ones, have V1, V2, have relatively small receptive fields. So now we've done a whole lot of work on visual cortex. We've gotten maps. We know something about the sizes of the receptive fields within these maps, and people are continuing along. So I should say that the next set of things that's going on is John Winnower in my lab, things going on in my lab. There's many things going on in other places, obviously. In my lab, Jonathan Winnower is just finishing up making measurements where we now know enough from the bold measurements that we felt we could compare them quantitatively with intracranial recordings from patients who are undergoing epileptic resections. And we have electrical signals coming out of these same pieces of cortex that we have bold from, and we're trying to close the gap between the cellular responses with which we measure with the electrical signals and the bold responses. And that story is very interesting. I won't do it here, but it's surprising to me. And Kendrick Kay, also another postdoc in my group, very, very smart guy, is been looking at the properties. I showed you the simplest first-order linear model for characterizing those population receptive fields, and it did okay for first-order stuff, but it had lots of little imperfections with it. And Kendrick has really cleaned that stuff up and has discovered a number of useful things. Okay, end of that section. I'm about to move from fMRI to tractography and so forth. If there's any questions about fMRI, this might be a moment. Sir.
I don't know how fMRI related it is, but you had that really neat animation of the visual stimulus where it's an expanding circle. And then it's kind of surprising for somebody who didn't know this, but you see something that looks kind of like an expanding circle on the visual cortex. So it kind of maps out, but it's kind of, it looks a little bit distorted.
Yes.
So it kind of made me wonder, when you see a perfect circle, where does that perception that it's perfect come from?
Yeah. You could be a psychologist, really.
Does the fMRI give you any insight into that sort of question?
Let me not answer your question, but comment on a part of it.
Okay.
That map is highly distorted, and that distortion is called the cortical magnification function. And the central part of your visual field occupies a huge amount of territory compared to the peripheral. So stuff out there, when you say, suppose I've got 10 degrees in the periphery, and you say, how much cortex does it occupy? It might be half a centimeter. And now if I take 10 degrees near the center, how much does that occupy? About 4 centimeters. So there's a big magnification, and that's what you started out observing. It's very observant, and that's true. And that function is very fundamental. And that function is the same in human and macaque and in many different species, and it actually reflects stuff here. The second part of your question is very deep, is how do you build a theory that moves from these neural responses to things that you experience? And that's the homeland of psychologists. And that is not one. That magnification is not something that you don't think things that are out there that are 10 degrees are really, really tiny.
Right.
And so how it is you derive experience from that is something that is not going to be inherent in that particular image and remains, I mean, in a way, that's the heart of most of what people really want to know about the neural responses, unless you're medical or unless you're just into measuring stuff. So it's a great question. You could have asked the same thing about how do I know it's face, how do I know it's this, how do I know it's that. So it's a great question, just I can't do anything for you. Okay, so now we're going to go from cortex into into the white matter pathways. And so this is a picture of what you normally see when you see cortex, but when a guy like me thinks about it, I think about these structures inside, which are the white matter bundles that take information from, now you can see these little bits of blue I'll point out, that they take information from one piece of cortex to another. This is kind of the wiring diagram. And the goal this next 10 minutes is going to be to tell you how it is we start to see the connections measured in this way of where these axons from some of the cells, not all the cells have axons that enter into it, but some of the cells do, and how do we measure those axons and where do we go? I want to say something I will show to you later, but I'm not proving to you now, is that the connections themselves, this is another difference between typical engineering practice and the human brain, is that usually when you have a computer and you have a cable that runs between two parts of the computer, you count on that cable as being stable, as not changing on you over time, and you design around that idea. In the human brain, these axons change over time. They change during development, and they change with use. So that if you have a set of axons that's used heavily for reading, during the years in which you become a good reader, the properties, the cells wrapping those axons change in their properties. And so the cables themselves change in ways that almost surely are important for your ability to perform a task like reading or face recognition or whatever it is. You might be doing that. So I'm thinking about retirement here, basically. And one of the things I'd really like to think about is systems that have not only changes in the synaptic properties and the connections between the transistors and so forth, but if you designed a computer in which the cables themselves would actually get more or less resistance and change in terms of their timing properties, I think that would be kind of, I don't know, I'd like to see a theory of that. Okay, so there's a picture of what they look like in the human brain. I want to, we keep jumping many scales of resolution. These are many centimeters here. When you zoom in and look at these things, this might be this cross section here is on the order of about 8 millimeters. Individual axons are often grouped together in the periphery. They are grouped together by connective tissue like this that sort of surrounds them, puts them in groups. And the tiny little dots in here, which are like submicron in cross-section, are the axons themselves. But they travel together, whole bundles of these axons. All the axons from some region all come on out and cluster together and go somewhere else. And that cluster is called a fascicle. And the kinds of things that we measure with this tractography are these fascicles, groups of axons, not individual axons. So, but since the brain is structured that way, it's not that every axon goes every which way at once, but they travel together. That gives us a chance. To look at stuff in the middle of the white matter. Okay, so how do we do that? Well, the idea is to use an old MR scanning sequence that's diffusion. That's the diffusion sequence. I won't go through the physics of it. Very simple idea and very beautiful and used routinely in clinics around the world, is if you say, well, how far can water diffuse in a bundle of axons like this, in a fascicle like this? Well, along the length of the axons, it's not much impeded by the membranes. It can kind of go in there relatively loosely. So it has a diffusion length, a diffusion constant that's not bad. And you can measure it. Actually, I particularly am fond of this thing because you can measure this diffusivity with, honestly, on units. The units of diffusivity are, square meters per time. And it's common to use square microns per millisecond because it ends up nicely. So you can measure it, and you can measure it in this direction, and then you can contrast that with how much it diffuses in this direction here. Along the length we call, I should have said we call longitudinal diffusivity. And perpendicular to it is typically called radial diffusivity. And you can go and you can measure this in 2, 4, 6, 8, 100, 200 directions to go inside of a thing. in the old days, and I think this is the right place to start when thinking about this, you might measure in a bunch of directions and then summarize the diffusion in a voxel by some kind of a smooth surface. The first one that was used was introduced by a guy I'm very fond of, Peter Basser, he used a simple quadratic, positive definite quadratic form, which in this case it's symmetric, and it's the diffusion forms an ellipsoid, and that's called diffusion tensor imaging. It's the summary of the diffusion in multiple directions using an ellipsoid. The mean typical difference for diffusion is the ellipsoid in this case. And he went in there and said, you know, let's start by using this form here. And that's been used very widely, and maybe some of you have heard this expression DTI. In fact, I'm curious, have any of you heard this expression DTI? Okay, the same ones who raised their hands at the beginning. Okay, well done. Okay, so, but these days, in the slightly more modern era, people are using much more complex forms, but it's okay to start with this, I think, in an audience like this. And the math of this is very straightforward and so easy to go with. And if you do that, you can see that here's a cross-section of a brain over here, and there's certain simple measures you can make off of this ellipsoid that have populated the literature and that we use to find tracks and think about them. This is one number. This is a single number to characterize this. When it's very spherical, we say it's very isotropic. So the fractional anisotropy is very low. So we'll color it as dark over here. When it's very oriented like this, we say it has high fractional anisotropy. So it's very bright. And you can see in the middle of the brain where the fibers cross and all the fibers are packed in there in the corpus callosum going from one side to the other, it's very bright. Okay, and here's another big track. And there's brain over here, just things are going around in a circle. They're not particularly one way or the other, and so that has low fractional anisotropy. And you can also paint onto it the direction in which it's going. If it's going right-left, let's color it red. If it's going up-down, let's color it blue. And back-forth, back to front, let's color it green. So now you can see why people would get excited about a method like this, because not only do we have the cortex, and not only do we have the white matter, but we can actually see something about, in the MR scans that you're used to looking at, here's the white matter, and it's all just white, for God's sake. Here, we're looking at the same stuff, and now we've color-coded it to say, where are those axons headed? Headed front, back, left, right? We can start to parse this thing up, and we can see stuff about the living human brain that we could never see before. And that's all within this technology has become commonplace just in the last dozen years. It's really it's that new. And it takes a while to get used to doing this kind of stuff. The other thing you can imagine, and people rushed into this, and it's one of those fields that I'm also very fond of, but still things are being worked out, is to say, well, I don't really want to have just voxel to voxel, because I know that in the brain they're connected, and they actually form these tracks and these big chunks. So this is an actual brain. This is the optic radiation. It goes from the lateral geniculus back to primary visual cortex. The eyes would be here. The primary visual cortex is here. It stops in the middle and comes back over here. And you'd like to be able to estimate those. Well, what you've got in the diffusion data as summarized from DTI are this whole bunch of ellipsoids like this. And when you see a bunch of them like this that are all pointed in the same direction, you'd say, well, they're probably part of a track, because we know that there are tracks in there, and this would be a good bet for a track over here. So there's a whole lot of math involved in the building up of these tracks and testing whether it's a track or not and so forth. And it's great stuff for people who are computationally minded and in engineering to work out. And this particular one was of great interest to us because we're vision scientists. So we can now go get estimates of these tracks. Here was the original brain, a sketch of a typical brain picture from somewhere from the literature. And these were our estimates, and we did comparisons between our ability to estimate this particular track and anatomical data and so forth. And so for this one, for the optic radiation, we're not bad at it, but boy, there's a lot of tracks in there. Some of them are bigger and some of them are smaller, and my confidence in our ability to get most of them is very low. So it's an area that I think is, for those who are suited to math, it's good. And I wanted to say something affirmative about all of these folks. Aviv Mezer is working on new methods for studying, new MR methods for studying the tissue properties in these tracks. Franco is studying all kinds of algorithms for evaluating tracks. Anyways, a whole bunch of stuff here going on. It's a very big open field, and anyone who wants to talk with me about it, I love talking about this stuff. Okay, so now we're at the final few minutes, and I'm going to give you a quick overview of 2 applications and then stop. The first one has to do with, as I said, I always look for applications, and one of the applications of vision is reading. If you can't see, you can't read. And I started to look at the development of reading and the vision's relationship to it. And when I started in the field, this is what, and this would have been about 2003, 2005, people were very interested in this piece. By the way, all of these are showing in this mode of resolution, which is older technology for rendering images. They're all showing the same actual piece of brain. And what they mean to be showing is this piece of brain right here. So here are the eyes. Here's the back of your head. This is the middle part. You're looking at the bottom of the brain, and you're looking at a little piece of brain right here. And but on all these guys, the best visualizations that they could do to show you what this looked like was to show it like this. And I'm sorry, I can't do anything for you about that. But that's just the way the visualization of this stuff goes. This is actually on the lateral surface where the actual thing is on the bottom. Anyway, now let me not complain about this. That was interesting. And the reason that was of interest was because of a series of papers in various ways showing that when children learn to read, changes happen in the responsive cortex over here. There's another paper that showed that when you had an epileptic subject in and you had intracranial implants here, 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 and here, when the subject read, there'd be activations from over here, but not from over here. There were a series of other things. I have 2 very long review articles going through this, but there's a lot of reason why people got interested in this piece of cortex. Now, I'm a vision scientist, so I see this piece of cortex over here, and I think, that's like my home base. That's right near all these other maps that I've been measuring. People who are in the field of reading, I don't know if this ever happens in your field, but people in the field of reading knew that visual cortex was around there somewhere, but they weren't exactly sure where it was, and they definitely didn't know where these maps were. They, well, okay, so let's go. So, you know, the simplest thing I could do, because it was easier for me than anything else, was to say, here's that piece of cortex right there. Where are the visual field maps? And you can see from my point of view, this thing is just buried right in and amongst a whole bunch of maps that I had just been measuring. It's like right next door. And so that was interesting for me to know. So I felt, okay, well, I could keep going. And in fact, it turned out, Andreas Rauschecker and I showed that this area here that's responsive to reading, in fact, also is it has a little map in it. So the things in the fovea are located over here, and things in the periphery are located over here. So the thing that you learned to train in your brain when you first started to learn to read was a little piece of cortex right in the middle of these visual field maps that's just kind of made available for you, to learn to recognize different forms, right there. Faces, by the way, there's a very large face responsive area just right next to it, just right adjacent to it. Areas for other expertise. So this area here seems to be a resource for learning to read. And when you pointed at me and said 2, that meant 2 minutes, right? Yeah. So you can, in fact, using functional MR, you can see if you put a word up there in the right visual field and you look in the left cortex, you can see that there's activation V1, V2, V3, V4, and so forth right in there. And you can sort of see the little sequence of activations going right through the cortex. And if you look broadly at the brain, where people have reported activations to reading, or for people who have poor reading and so forth, you can see that they're spaced like this. So somehow these signals that start out in visual cortex must be getting here. And using the diffusion methods, we can start to look at the tracks that connect these. The blue one here is a particularly famous one called the arcuate. And it turns out that the tracks themselves, not shown here. Another one, here's the blue one is the arcuate. Here's another one that goes from the visual cortex on forward is called the inferior longitudinal, ILF, whatever, inferior longitudinal fasciculus. In fact, you can actually measure the fractional anisotropy on this track here and how it develops in children and measure their reading. This is a crazy graph, just insane, right? This is how water is flowing back and forth in certain directions along this tract here. And here's how well they scored on standard reading tests over here. And you can find a correlation with an R.51 between the two of them. It's just bizarre, right? And that's why I was saying to you, wow, the wires are themselves developing, and the way in which the wires develop actually has an impact on reading. In fact, you can look just at the wires. I'm now going to tell you about measurements on this blue track and this one. If I take the fractional anisotropy, or this track and this track, and I try to predict reading, so I predict reading from the fractional anisotropy on this track and this track, I get a great prediction. It's actually a pretty good prediction, yeah.
So do you have a similar graph with blind children with a different set of wires going from, say, somatosensory areas?
Right. I don't,
Okay.
but people do. And people look at both blind, late developing, people who have not, let me draw your attention, people who have not ever get trained, illiterates, things like this. So the field is burgeoning, and I'm a very, very small part of that field.
Are the reading tests done before and after the fMRI, or just before?
These are our data. Yateman is a guy in my lab, and these are our data. And we followed these kids for 4 years, and we did reading tests every single year. And one of the things I don't feature here, but it's a little disappointing to us, if you take the rank order of how well the kids did in their first year, and then how well they did in their fourth year, it was essentially invariant. And you could actually see this in the development of the tracks in certain ways. But anyway, we described that in the paper. But we measured it repeatedly over time. I didn't mean to defend or even describe all of the things about reading, which I'm happy to talk about next. But I wanted to do one example for you of how these different things come together. The functional signals, the diffusion signals, to start to piece together a story about the whole circuitry in the living human brain. And that was really the goal of that. And I'm not going to get to the surprise, but there's a whole story here that I'm going to skip, but I'm out of time. And I wanted to say that the neurologists keep turning up these cases that I just find amazing. And I just wanted to thank, I got started in this work with Dave Rummelhart and Gary Glover. Some of you might know Dave or Mike Shadlin. And I wanted to thank these are all the folks from several years over the lab. I work closely with Bob Doherty here, who some of you may know. Sergio Milan was population receptive field modeling. This is Peter Catrisa, who did nothing at all on MR, but is known to you because of his, but he was part of our lab and worked really on the digital imaging work. It was great to have in the lab. And I've been very lucky to have a set of visitors from Japan, who's shown here. These are all ophthalmologists except for John Winnower. And they've been extremely helpful in identifying patients and subjects that helped us build up this story. Oh, and this is Joyce Farrell. She's my wife. And so thanks to all of you for your attention.
We have time for one question.
I would ask, if we look at MEG as an alternate way of tracking some of the same sort of things, does it have useful correlation?
I'm going to tell you the thing that has me stunned. We've just done these intracranial implants, and then we've put EEG, not MEG, but EEG on the scalp. And the signals that are driving the this fMRI response are that you measure with EEG are completely ineffectual at the bold. It's like they're complementary signals. It's like you're getting two independent measurements out of cortex as near as I can tell. And I haven't published it, and I haven't defended this in a tough-minded audience and so forth. But we're staring, in my lab, we're staring at this thing and saying, well, those electrical signals, okay, they're really big. Those bold signals, they're really big. They seem to be coming out of different aspects of the circuit response. So that's either good.
Or the MRI field is causing the bold signal to alter the EEG.
Well, that we're measuring them at different times. We take the same person and put them once in EEG and once in bold, and so there's no interaction between the measurements.
That you get from the bold or from the MRI could be a function of what you would get without the field there, plus you're superimposing this rather stimulatory field.
Well, we'll think about that one.